All right, well, I just want to thank the people for inviting me to come and talk to you today. So as Mark has indicated, I come from University of Manitoba. I was previously at Dalhousie University, and so I've been looking and studying different forms of ovarian cancer for about the past 20 years or so. And when I entered the field, really there was pretty much nothing known about ovarian cancer other than it was really bad and women were dying from it at alarming rates. And in those 20 years, there hasn't really been much change in that fact. And so, yeah, I'm just looking for the old slides. And so, what I'm going to tell you today is really about some of what we've come in those past 20 years, but really kind of what's going on right now at the forefront in this particular area of this type of study we're doing on something called chromosomal instability. So I have nothing to disclose with regard to this project. I just want to highlight, of course, some of the people that are involved in this. And this really came out of a collaboration. So I'm not a chromosomal instability person by training. I'm not even an ovarian cancer person by training, but it's what I've done for a long time. Really, this came out of a collaboration with this gentleman right here, Dr. Kirk McManus, who is a chromosome and genomic instability expert. And this is a collaborative project that we do, and I just want to today highlight a couple of the people, Chloe LePage and Claire Morden, whose work is really right now driving this field forward, specifically with regard to high-grade serous ovarian cancer. I think you'll all recognize this fella right here. He's the one that actually, got me to come and join this group and basically said, Mark, you're doing this thing and come and teach. So with our group in Manitoba, really, Alon is just highlighted here, but he's really representing all of the gynecologic oncologists, all of our fellows, and as well as the residents that are critical for a collaboration, because all the work that we do is with clinical samples, so that we're trying to get the best idea as to what's going on with the patient, right from samples from the patients. We don't typically use cell lines for any of these types of things. These are all primary patient samples. So it's with the cooperation of all the people like you that is really driving this forward, so that hopefully what we'll get to is to be able to understand the disease better, to be able to create better therapeutics to improve the health of women with these diseases. So one of the fundamental priorities that was identified in the 2015 Ovarian Cancer Action Meeting was to characterize genomic instability in drug-resistant high-grade serous ovarian cancer. And really there's two features there that I want you to focus on, and that's the whole notion of genomic instability and drug resistance. And that's really what I'm going to be talking to you about today, and how we actually arrived at that point. So the learning objectives you've all seen. Really, we want to look at, I want to tell you about the fact that there are different kinds of genomic instability. This is a word that's been used, and it's used interchangeably with actually quite different genetic events that occur. And they're actually very distinct processes. They're very distinct mechanisms of action, which means we should be targeting these things quite differently. And in fact, within the spectrum of ovarian cancers, the reason that our cytotoxic agents are ultimately failing is because of different kinds of responses to genomic instability. We're actually not targeting the disease properly. And so the hope for the future would be that we would be able to develop therapeutics that can target each of the different histotypes of ovarian cancer more specifically to have better outcomes. So today we're going to go through some of the different things, like looking at important characteristics that in the chromosomal changes in high grade, as well as looking at different aspects of chromosomal instability. Okay, so this is a timeline of typical treatment for a patient with high-grade serous ovarian cancer. And the reason I'm focusing on high-grade and not type 1 tumors is because, one, it's the most prevalent. It's also the one that we have the most samples on, and it's really where we thought we would make the best inroads. So with this particular patient, what you'll see is she was a poor surgical candidate initially, so started neoadjuvant chemotherapy, a couple rounds of cytoreductive surgery, finished her chemotherapy, recurrent disease, no longer could tolerate paclitaxel, so they gave her doxorubicin, carboplatin, Eventually recurrence, again another round of chemo, throw in some tamoxifen for, just to see if that would do anything, recurrence again, and subsequently death. So my first question to you is, and now we'll flip over to question 1, is why does the cancer keep coming back? So that should be on your app. You should be able to answer this. Hopefully the multiple choice questions will come up. You should be able to see those. If not, it's okay. I could just read them out. So cancer cells can reduce uptake or increase efflux of chemotherapeutics leading to chemoresistance. That would be... Oh, there we go. So is it that first one? Is it that cancer cells can subvert apoptotic mechanisms? Is it that cancer cells can repair the damage caused by chemotherapy? Is it that cancer cells can exhibit cellular heterogeneity in a tumor such that you've got a completely different populations of cells, some that are sensitive and some that are resistant? And is that actually all of the above or none of the above? So feel free to select your answers. We'll give you a minute to do that.
You got that? Where can we find the app then? I don't have it in my...
Or we could do it by show of hands.
Yeah,
we
let's
could
do
use
it.
the old technology. So who
you don't
thinks
have
it's...
to be...
It's any specific one? Who thinks it's A, B, C, D, all of the above? Oh, good. Okay, none of the above. If none of the above, you should go back to rudimentary. So yes, all of the above, except the part of the point that I'm trying to make is, would you be surprised if the first 3 are actually all pretty much a function of the 4th one? And it's this whole notion of tumor cell heterogeneity. Tumors aren't one particular thing. They're a mixed bag. And often they're a mixed bag with quite different genetic characteristics, as well as a mixed bag of metabolism and whatnot. And it's really that whole idea of, look at 95%. Who's the one holdout? Okay. Okay, well, let's go back to this. Okay, so this idea of tumor cell heterogeneity and drug resistance. So for this particular slide, what I'd like you to focus on more are the colors. So let's say we've got a typical diploid cell, something bad happens, it transforms, and it's got 2 daughter cells that are both viable, but now we're starting to get a little bit of genetic changes in these, and so just the color indicates that they are different. But let's say in this particular cell, now you've got a cell that has some drug resistance to it. Okay, well, each of these are going to continue to multiply, divide, and now you're going to get a whole panoply of different cells, some of which, when they have genetic changes, they're no longer viable, so you don't see them as present in the tumor. Some additional ones will have drug resistance. So when we start adding all these up, we get these solid tumors that are this big mixed bag of cells. Well, in our typical treatment with cytotoxics, you've got your cells and the vulnerable cell populations that'll die in response to those cytotoxics, but you're left with often cells that are now become drug resistant. And so that they'll continue to proliferate, and because we've got this kind of continuous genetic changes going on, what happens is when tumors recur, now you've got basically a completely different disease. So we keep throwing the same cytotoxics at it. And eventually, yes, you will just select for cells that are no longer sensitive to those cells, and that's how recurrence as well as drug resistance is actually selected for by the treatments that we're giving them. So how does these cells become genetically diverse? Well, the underlying theme for this is something called genomic instability. And really, genomic instability is just the state of increased DNA mutations, copy number changes, and epigenetic alterations. And those all play a critical role in cancer initiation, progression, evolution, and drug resistance. You can imagine from cell to cell, if you've got completely different DNA, different genes being turned on, some have gained oncogenes, some have lost tumor suppressor genes, but you've got a completely different set of cell populations. And that's all caused by genomic instability. But what is genomic instability? So again, this is a term that it's an umbrella term that covers very distinct types of changes. And so, for example, microsatellite instability is one type of genomic instability. And typically in tumors, you'll actually see that many of these are mutually exclusive. You tend not to see microsatellite instability in some cells in tumors that have chromosomal instability, for example. You sometimes can see some of these features, but they're not common. So tumors that have microsatellite instability are characterized in patients with Lynch syndrome or the endometrioid and clear cell ovarian carcinomas, for example. And microsatellite instability really reflects defects in mismatch repair genes, as well as hypermethylation of DNA promoters, so that genes are typically turned off. And you can imagine if you've got hypermethylation of tumor suppressor genes, now you've effectively lost those. So if you sequence DNA from a cell, you won't see that, however. So you'll need to specifically look for methylated DNA patterns, for example. So MSI is a type of genomic instability not to be confused with the other types. CpG island methylator phenotype, or the CIMP phenotype, is another type of change. And again, that's where, okay, sorry, I just completely screwed up the MSI, didn't I? That's because I'm getting ahead, I'm too excited. So you don't see hypermethylation in MSI, pardon me. MSI is further characterized by either expansions or contractions in microsatellites. That's why it's called microsatellite instability. CIMP is where you're seeing the DNA methylator phenotype. So that's where promoters get hypermethylated and essentially genes get turned off. And then chromosome instability. This is the least well studied and the least well known of all the different kinds of genomic instability. It's best characterized in colorectal cancer, whereas up to 85% of all colorectal cancers actually have chromosomal instability. And frankly, when we apply this to ovarian cancer, we really don't know what the rates or percentages are. However, and that's because people have never actually truly measured chromosomal instability. What all the DNA sequencing that's been done has done is it's indicated genomic instability. But that's really because they've looked at populations of cells. To look at chromosomal instability, you have to look at single cells. And we'll tell you why in a moment. So chromosomal instability is just beginning to be learned about in ovarian cancers. And again, we're simply focusing initially on high-grade serous ovarian cancer. So why do you care? Why should this group even care? Well, it's because chromosomal instability is associated with cellular transformation, tumor evolution, metastasis, drug resistance, and ultimately poor patient outcome. And so really we want to know is what causes chromosomal instability in these patient populations, and does this mean it's now going to be an opportunity to develop targeted therapeutics that might be able to target this process? Because if really we're talking about chromosomal instability as generating cellular heterogeneity, if we can target chromosomal instability pathways, we may have a whole new kind of broad spectrum of treatments. So SYN also comes in two particular flavors. It comes in numerical and structural. And really numerical is just a change in the number of chromosomes. That can be gains, that can be losses, it can be parts of chromosomes, what have you. So in this, comparing a typical diploid cell, you'll see that this cancer cell has, I didn't even bother counting, lots of extra chromosomes. This is a cell that's dividing in the patient. I don't think I would want this cell growing in my body, personally. But this can also reflect losses sometimes too. So now, just so you know, in most cells will tolerate gains in DNA much easier than losses in DNA. And so those gains are what we typically record in high-grade serous ovarian cancer. You can also have structural, and really what this is just reflecting using chromosome paints is translocations within chromosomes or parts thereof. So if we take a look at this particular cartoon, if we take a look at this cell in the middle being, if you will, our normal diploid cell, just characterized by two individual chromosomes of these three different colors, you can see that numerical SYN can be viewed as small-scale gains in chromosome numbers, for example, or parts of chromosomes, large-scale gains, such as complete polyploidy, or small-scale losses, parts of chromosomes. whereas structural sin can include deletions, amplifications, inversions, and translocations. And in some cancer cells, you can actually see both numerical and structural sin, which is fairly typical. Once you have a genome that's unstable, it's going to continue to be unstable. And chromosome instability is a rate of continuous change of gains and losses. And really, that's what we're having to measure. But that's why just sequencing a tumor won't tell you that. You need to look at the single cell level. So I'm going to show you how we actually measure chromosomal instability. So just again to reiterate, sin may induce simultaneous and ongoing changes in copy number changes in large cohorts of genes. If you have an entire arm of a chromosome, you've changed a lot of genes. So that can include oncogenes, tumor suppressor genes, DNA repair genes, and apoptotic genes that promote oncogenesis. Essentially, what sin induces is randomness in the genome, so that every single cell may have a completely random genome, as long as it's viable. So just like that previous diagram where I had you kind of focus on the colors, now I'd like you to focus on the number of chromosomes in these particular cells. It's because sin is associated with drug resistance and tumor recurrence. So if here's our diploid cell, you can see there's just 2 copies of each of these chromosomes. Maybe once they divide, you have some chromosome loss in this, which produces a drug-resistant variant. This one has a bit of chromosome gain. They continue to divide, and you've got all of these different changes that are reflected by the different chromosome numbers. That produces that heterogeneous cell, which again gives you the same scenario. But it's because they're so genetically diverse that they're just no longer responding to our cytotoxics anymore. So sin promotes multi-drug resistance by creating that heterogeneity with greater levels of genetic diversity. So it can circumvent oncogene addiction. So oncogene addiction would be where you have a particular cancer which kind of arises and relies on one particular oncogene that's driving that cancer. That if we now have a targeted therapy that specifically targets that particular oncogene, you can kill those cells. And you tend to see effective therapies with targeted therapies that do that in some of the less genetically complex diseases, such as some of the hematologic diseases. But ovarian cancer is so genetically diverse, they just don't work. And that's why even targeted therapies eventually will fail as well. So sin may contribute to decreased efficacy of those targeted therapies because of the genetic diversity. That's really the message that I want you to be able to take home today, to understand that the diseases you're working on are very genetically diverse and they continue to evolve, if you will. So what causes sin? So the whole study of sin actually came out of yeast work. And because we have the entire yeast genome sequenced, we know exactly how many yeast genes cause chromosomal instability. So what Kirk did was using his information from yeast, he started to look at human cells and look at the human genome. Now, currently, there's only about 150 sin genes that have been identified in the human genome, and not all of them have been absolutely proven. However, based on the genome, there's an estimated 2,500 sin genes in the human genome, which means there's a lot of work still left to do to identify the pathways and the targets that contribute potentially to sin. So what can cause sin? Well, chromosomal segregation defects, DNA repair and replication defects, cytokinesis defects and endoreduplication. And I've just put up this picture of a cytokinetic defect from a sample from ascites. And I hope you can actually see this. While there are some cells with a single nucleus, this is one cell here. It has 27 nuclei. This was what we would consider chromosomal instability. You now have 27 nuclei, so 27 copies of every single chromosome, and probably every nucleus is different. That's going to set up something so that this patient, and we know, didn't respond well and developed drug resistance rather quickly. Now, there can also be cell cycle defects. So all of these together basically mean you're getting different genetic changes. But there's also some non-intuitive pathways and targets that we've identified that can contribute to sin. That can include lipid or tRNA biosynthetic pathways and proteasomal degradation. So, for example, a sin gene is cyclin E1. Cyclin E1 is upregulated in 20% of all high-grade serous ovarian cancers. It causes chromosomal instability. And what we're actually studying is the proteasomal degradation pathway for cyclin E1. It's maintained, and what we found is a lot of the genes that are supposed to degrade it are actually affected in high-grade serous. So those are some of the non-traditional targets that we're looking at currently. Okay, so how do we go about studying this? I've already kind of gone on to say, well, we're not sequencing full tumors, we're looking at single cells. So we can do this in a couple of different ways. Fortunately, with our clinical collaborators, we have a tumor bank where we get solid tumors as well as we get the paracentesis. And so because from the paracentesis we can actually isolate and grow the cancer cells, we now have essentially cell samples that we can look at these changes at individual cellular level. One of the simplest ways of determining if you have SYN or not is to count chromosomes. So again, here's just a diploid cell, and we can do metaphase spreads to look at cancer cells. So this is one particular cell from one of our cancer patients. But you can't just look at one cell. You have to look at hundreds of cells from the patient to know whether or not you have heterogeneity and whether or not SYN is an actual feature. And so that's typically some of the things we'll do, and I'll show you some of that data in a moment. But because it's really laborious to take a cytogenetics effect, Kirk wanted to actually see if he can modernize this. And so using imaging analysis that he developed, essentially what we can do is we can actually look at nuclei, and that's what each one of these little gray blobs are, is a nucleus, and look at its nuclear area. So you can imagine if you have large-scale gains or losses, the size of your nucleus actually changes, and that's actually correlated with the number of chromosomes. So what he was able to do was just using a high-content microscope, we can count nuclei. Here in the normal, you can see most of the nuclei are about the same size, same shape. But in this cancer high-grade serous patient, you can see the nuclei are different sizes and sometimes different shapes. So we've now can automate this so that our data will reflect this. Here's just another patient, however. You can see this patient was a poor surgical candidate. So she didn't get any surgery. They tried to treat her medically. First with paclitaxel carboplatin. Upon recurrence, she didn't do well, so calyx, but she died. So this is a very aggressive advanced case patient when she came in. What these letters represent are all the paracentesis taps that we got. And so what we were able to do was to actually measure nuclear area from each of those paracentesis taps. And what I'll just point out is over time, our nuclear areas are very dynamic. So there's continuous genetic change within this particular patient. Not only that, if we count the number of chromosomes, and actually we have an algorithm of which we have developed something called a SYN score per nucleus, and this is typically 200 nuclei per sample, or a minimum. What you'll see is that a SYN score of 0 in ours is actually a diploid cell. And so before she got any treatment, she was actually fairly diploid. As the disease progressed, you can see the dynamics continue to change. And this reflects both gains and losses that work into our SYN score. So basically, the message here is SYN is dynamic over the course of disease progression and potentially in response to treatment. So another way that we can do this is to actually, because we want to automate some of these things, is we can actually just use fluorescent in situ hybridization, or FISH, using different fluorescent colored probes that will tag chromosomes. In this case, we've got red, green, and yellow tagging chromosome 8, 11, and 17. And in this picture, the blue stuff is the nuclei. And in a diploid cell, what you should be seeing is 2 copies of each of these chromosomes. And indeed, that's what we see in this. We see 2 green dots, 2 yellow dots, 2 red dots. So this is a diploid cell. We can now take this technology and use it to look at cells from paracentesis across the disease spectrum. So we now have an automated method of doing this. And part of the reason for going toward this automation is to see whether or not we can actually develop SYN as a potential prognostic for patients. What we're more excited about, while we've applied a lot of this technology to paracentesis samples, we now have been able to transfer this to archival tissue samples. So here's archival high-grade serous ovarian cancer tissue. We're looking at chromosomes 8, 11, and 17. And In this particular image, what you'll see is we see a nice diploid cell in part of that sample, but right beside, we see an aneuploid cell, so a cell that's got different genetic diversity. In this case, it's got 11 copies of chromosome 8. So because we are now getting this toward more of an automated, we can now look at archival samples, and the whole point of doing that is so that we can now say, is SIN associated with stage, grade, drug resistance, patient response, and patient outcome? Because our whole point is to drive this forward so we can learn more about the disease so that we might be able to use this as a prognostic, as I've indicated. Okay, so question 2. Can platinum drugs and taxanes cause SIN? So remember, if you go to that timeline, we saw that SIN was very dynamic. Was that because the disease evolved? Or is that because we were selecting more SIN cells because of our treatments? You guys are so smart. But they're kind of obvious questions, so I figured you guys had exams this morning, you needed easy questions this afternoon. So the short answer is, if anybody wants to change, the short answer is yes. As a matter of fact, if you think of the mechanism of action of platinum drugs and taxanes, they are perfect at screwing up DNA, and actually they are touted as being perfect drugs to cause SIN. Well, is that good? Should we be causing SIN? So let me just show you what, again, that same samples before I get to that idea of should we be causing SIN. This is sample B. So if you take a look at sample B, these blue things are the nuclei, and pretty much all of them are diploid. If we look later after treatment, now what you're starting to see is a very different picture from this same sample. As a matter of fact, this is one nucleus. So now you can see there's differences in nuclear size, there's differences in nuclear shape, and I think in this one nucleus, you can see that there's lots of copies of different chromosomes or fragments thereof. So our drug treatments correlate with greater SIN as well in these patients. So is that good or is that bad? Well, we want to take advantage of SIN in these tumors for a reason, and it's so because we can take advantage and use it therapeutically. So part of what we talk about in this is something called a SIN threshold, which really reflects the gains and losses. And as I said, cells can tolerate gains better than losses, but at some point you reach to this SIN threshold. And if you have a therapy to drive SIN beyond that tolerable threshold, the cell should die. And really, that's what the cytotoxics are doing. They're driving them beyond a SIN threshold. But eventually, because we've now got such genetic diversity, you've now got cells with drug-resistant mechanisms. So part of what we want to do is we want to continue to exploit this feature, which is common to all high grades. We don't know about the type 1 tumors yet. That is part of our next steps. So that we can exploit that for future therapeutic application. And that's why we need to understand part of the mechanism. That's why we need to identify these novel SIN genes so that we might be able to use them in a synthetic lethal approach to try to kill these cells. But we're not going to be going after one specific oncogene or tumor suppressor gene. We're going to be going after pathways. And that's really where combination chemotherapy will kind of be the hope for the future for these kind of patients. You've got very genetically diverse tumors, and single monotherapies or even the combinations that we're doing, while they give us some hope, we're going to have to do better in the future to develop additional combinations. So with that, I'm happy to take any questions. Thanks for your time.
Thanks very much. Any questions? All right, we got one.
Question
Sorry,
in the
yeah,
back?
Thanks. Michael?
I don't know if I heard the answer to the question. Is creating SIN bad?
So the answer to that question is creating SIN is probably good if we can take advantage of it by targeting, once we induce SIN, if we can push it even further over the edge, that would be a good thing because it should drive all the tumor cells into a cell death. However, the problem is our current panoply of drugs doesn't allow us to consistently push cells over the SIN threshold.
And the chemotherapy effect on SIN, is it dose-related, time-related?
So those are all excellent questions that we hope to answer. Yeah. Great. Thank you very much.
That's great. Thanks a lot.